Hello children. So we are moving to the second topic of our chapter, that is the pre-fertilization structure and events. Topics which are focused for today's class are structure and development of microsporogenesis and microsporogenesis. So children, before moving into the details of the structure and development of microsporogenesis, you have to be thorough with the structure of the stamen. Here you can see three pictures of stamen, where in the first picture you can see the stalk-like structure filament and the two anther lobes. Second picture you can see the connective also, where the anther lobes are connected to each other with a sterile tissue known as connective, and you can see the stalk-like structure filament also there. Now just focus on the third picture, where the anther lobes are cut into two and you can see four chambers there. Since they are two-lobed structures, bilobed structures, there are four chambers. And just focus on the word written on the top, that is dithecus. Theca means the chamber. Since it is bilobed, they are having two chambers, and total you can see four chambers. Since it is bilobed, they are having total four chambers there, and you can see the release of the microspores or pollen grains from the microsporangia. Now, as you are clear with the structure of the stamen, we can move to the structure and development of the microsporangium. The pollen grains or microspores are produced within the anther of the stamen, that is the male reproductive organ of a flower. A typical anther consists of 4 microsporangia. This type of anther is said to be dithecus. As you learned, dithecus, di means two, theca means chamber. The stamen is developed as a projection first, and then it elongates and the tip becomes four-lobed structure. This four-lobed structure is the young anther, and each lobe contains an elongated microsporangium. Now you can see a picture of development of an anther, a diagrammatic representation. It is having A, B, C, D, E, F, G parts. Okay, so we'll begin from our explanation will begin from the A part. Here you can see the four-lobed anther begins its development as an undifferentiated mass of compactly arranged homogeneous parenchyma cells which are surrounded by an epidermis. First it become two-lobed and then it become four-lobed structure. In the diagrams D and E, you can clearly see the four-lobed structure of the anther. But it begins its journey from an undifferentiated pattern, which is represented in the picture A. There you can see the formation of homogeneous parenchyma cells. That means almost same pattern of arrangement of parenchyma, simple parenchyma cells which are surrounded by an outer layer epidermis. Then within the four lobes, some of the hypodermal cells at each corner become differentiated by their large size, radial elongation, and more conspicuous nuclei. These form the archisporium. You can see the archisporial cell in the diagram B. There you can see at the corner, each corner, those cells, those parenchymatous cells are becoming differentiated and they are increasing their size, they are elongating, and they will have more conspicuous nucleus. And they forms these kind of changes, convert the cells into archisporium. And each archisporial cell then divides to form primary parietal cell towards the epidermis, that is towards the outer layer, and the primary sporogenous cells towards the inner region. You can see the third diagram where you can see the primary sporogenous cells towards the inner sides. Primary parietal cells towards the epidermis and primary sporogenous cells towards the inner region. Then the primary parietal cells which are dividing towards epidermis will divide periclinally and also anticlinal divisions. They will undergo periclinal and anticlinal divisions, and they will form the wall of the anther. You can see two types of divisions here: sporogenous cells, primary sporogenous cells, and primary parietal cells. Parietal cells divides outwards towards the epidermis, and sporogenous cells divides towards the inner region. And those cells which are moving towards outside, primary parietal cells, they will undergo again and again anticlinal and periclinal divisions, and they will form the wall of the anther. Just focus on the C diagram, and after that you can see the primary sporogenous cells divide repeatedly, form sporogenous cells. See the term sporogenous, spore generating tissues. Those cells will form a group of those group of cells are known as the tissue, and it will form the sporogenous tissues. The sporogenous tissues occupies the center of each microsporangium and is composed of sporogenous cells. Sporogenous tissues, obviously they will be composed of sporogenous cells, which are also known as microsporocytes. The sporogenous cells have abundant cytoplasm and prominent nuclei. These are the specialities of the primary sporogenous cells and primary parietal cell. Parietal cells will form the wall of the anther. Now you can see the D diagram there. There you can see 4 layers which are forming the anther wall. That is the primary parietal cells from the diagram C. They divide anticlinally and periclinally, and forms the outer layers of the anther, and towards inner region sporogenous tissues will contribute the sporogenous cells. Now you can clarify these diagrams with the explanation given in the next slide. Now here whatever we had discussed in the previous diagrammatic representation, the same explanation is given in this slide. That is how the cells are dividing initially, how the cells are dividing and how they are forming two-lobed to four-lobed structure, how the archisporium is formed, after that how the primary parietal cells are dividing, how the sporogenous cells are dividing and they are forming the sporogenous tissues, sporogenous cells, that is the microsporocytes, and how the anther is forming their wall, how it is becoming layered. All the explanation which we already discussed along with the diagram is given in this slide. You can just refer it. Now here you can see the structure of the microsporangium, how the young anther is maturing, how they are forming into four layers, how they are getting dehisced. Everything is represented in this diagram and in this slide. So you can see the wall of anther is multi-layered. They are composed of four layers. As shown in the picture, you can see the outermost layer is the epidermis, and then comes the endothecium. Inside endothecium, you can see the middle layers. Few layers are there. After that, you can see innermost layer, tapetum. Tapetum is the nutritive tissue which surrounds the sporogenous tissues and nourishes the developing pollen grains. The developing pollen grains consume the products of middle layers and tapetum. Cells of tapetum possess dense cytoplasm and generally have more than one nucleus. So here you can see the middle layers and the nutritive layer, that is tapetum, and they are nourishing the developing pollen grains inside the sporangium. And these spores, these nourished spores, later becomes the microspores inside this bag-like structure inside the sporangium, and total structure is known as the microsporangium. Now once the structure and the development is clear to you, just go through the picture once again. Now you can understand it better. That is, the picture A is representing the undifferentiated mass of cells which are turning into four-layered structure. Coming into the E diagram, you can see clearly the four layer, that is outer epidermis, then comes the layer endothecium, then comes the middle layer, after that inner, each corner you can see the tapetum, which are providing nourishment to the growing pollen grains. Okay, one enlarged portion also shown in the diagram F, and the last diagram G, you can see the dehiscing anther and the pollen grains are releasing. And you can compare the previous diagram also. This diagram and the previous diagram, you just do a comparative study. You can understand the concept better. Now, as we discussed about the formation and development of the microsporogenesis, structural details, and we discussed how they are developing. And now we are moving to the next topic, microsporogenesis. That is the process of formation of pollen grain, that is a microspore, from a pollen mother cell in the microsporangium of an anther is called microsporogenesis. As we discussed, the sporogenous cells divide and give rise to pollen grain mother cells or pollen mother cells or microspore mother cells. Pollen mother cell contain two sets of chromosomes and therefore they are in diploid state, that is 2N condition. As the anther matures, each microspore mother cell undergoes meiosis to form 4 haploid microspores, that is pollen grains. As meiosis, the reduction division takes place, the chromosome number is reduced by half, that is already it is in 2 set, now it is being reduced into 1 set, and the pollen grains are haploid in nature. The cluster or the group of 4 pollen grains is called pollen tetrad or microspore tetrad. Since we call the pollen grain as microspores, it is also known as microspore tetrad. Later, the mature pollen grains separate from this tetrad and get released as pollen grain.